Hi everyone. Just give me a second. I gotta switch windows here. Okay. Hi everyone. My name is Matt Penticuff, and today I'm going to be discussing a paper titled "Dynamic Reorganization of Striatal Circuits During the Acquisition and Consolidation of a Skill." If you were in class yesterday, you saw I think it was like the first 5 slides of this that we will be going over again in this presentation. A word of warning, the software in which I'm using to record this is OBS, or Open Broadcasting Software, and it is very finicky on a Mac, so you will probably have to see me do cuts. I have to pause it and then cut it to make sure when it crashes I don't lose the entire thing. That being said, let's continue on. So again, I wanted to start with a little bit of background on how we define a skill, basically for the context of this paper. It's highly generalized and divided into 2 sequential phases. The first initial phase being a rapid improvement in performance, followed by a later second phase of slow gradual improvements as a skill is consolidated or asymptotes. These 2 separate steps are important because previous studies have suggested that each phase may use different striatal circuits. The main focus of this paper is to prove that separation and discover how these 2 circuits differ on a functional level. So when we think about this, when we think about the 2 phases of skill learning, the best way in which I've thought about it is how piano, I think they actually use this in the paper as well. When you first begin learning a piano, you learn the notes in individual keys, but you very quickly are able to put down melodies and stuff like that. You see a nice rapid increase in improvement, but once you start mastering it, you can do small, subtle differences. You can get better, but it's not that huge leaps and bounds better. So that's one way to look at it. And you have to forgive me, I'm not used to using keynotes, so I'm trying to figure out everything as I go. Okay, so next I want to also provide a very simple overview of the striatum. I'm sure we've all kept up on the reading, but just in case you're behind, the striatum is a subcortical part of the forebrain that receives glutamatergic and dopaminergic inputs from various brain structures. Very generally, it could be divided into 2 subregions, the ventral striatum and the dorsal striatum. In the context of this presentation, we will be focusing entirely on the dorsal striatum. The dorsal striatum can then be further divided into 2 subcategories, the dorsal medial striatum, the DMS, and the dorsal lateral striatum. The DMS receives much of its input from the associative cortices, while the dorsal lateral striatum receives much of its input from the sensory motor cortices. It is at this level of differentiation where previous research has thought that the different phases of learning kind of separate, with DMS being primarily involved in early phases of learning and DLS being involved in more later phases of learning. So for the first experiment, the experimenters wanted to prove the separation of phases between the DMS and the DLS. To do this, they implanted electrodes bilaterally into the DMS and DLS to measure neural activity of mice learning a motor skill in the form of a rotating rotor rod, or an accelerating rotor rod. In this bottom right corner, we can kind of see the exact locations in which they implanted the electrodes. I don't know if many people can make sense of that. But the gray dot being the DMS location, the black dot being the DLS location. For the rotor rod, though, I don't remember if we actually went over this in class. I can't recall. But just in case we haven't, there's a little short video I wanted to show. So basically, a rotor rod is exactly what it sounds. It's a rotating rod, which you place mice on, and it's considered a motor skill learning because generally it increases in speed. So the task in which they are learning is, I need to increase my movement in order to stay on top of this rod. And eventually the trial ends at the conclusion when they either fall off that rod or at the conclusion, in this case, of 300 seconds. So if you look, they eventually just kaplunk. Yeah, it's nothing really spectacular, but it is what it is. So with that context, boom, boom, boom. So in that context, they were placed on this rotor rod for 8 days with 10 trials a day, and the rod would increase during these trials from 4 rotations a minute to 40 rotations per minute. So this next part seen here at the bottom part of the page is kind of pretty important. It defines what the experimenters defined as early and late training phases. So for the first two trials of day one, we're considered the naive phases, are naive trials. So basically, the rats had no skill or memory of a skill of what this rotor rod is. It's basically very fresh to them. The last two trials of day one were the early phase of learning, where you are expected to see that nice rapid increase in performance. And the last two trials of day 8 were considered the late phase of learning, where you see small gradual improvements. So with that, so what exactly did these electrodes measure? So there were two basic metrics that the experimenters used: the rate of modulation, which is basically a change in firing rate of a change in firing rate from a baseline, and then the percentage of task-related neurons in these locations that changed firing rate during running. So for this, in terms of the task-related neurons, there wasn't a significantly or a statistically increase in firing rate for the DMS, even though it is larger. Statistically, it wasn't enough to be labeled significant. But for the DLS, they did see a nice increase from 74 to 80 to 89. Positive modulation increased during early training for DMS and during late training for DLS. Now, positive modulation is basically an increase in firing rate. Modulation can, well, actually, I think it's on the next slide. It'd probably be easier to say. So if you're looking here at this bottom part, there are two types of basically ways in which you can modulate a neuron. You can do a positive modulation or an increase in firing rate during running, or a negative, which is a decrease in firing rate. So it kind of sets that paradigm of DMS being involved in the early phases of learning, where we see a nice positive increase during this early phase, and then the DLS being more involved during later phases of learning, where we see that nice increase during late phases. The top one is just a more basic, but also not insignificant figure showing the rate of modulation overall. Again, it follows again with that paradigm of early being DMS, late being DLS. So we see now through these neural measurements that there is a difference in neural activity during the separate phases of learning in the DMS and the DLS. So if we were to lesion these areas, how would this affect performance? Would it follow with what we would expect? So when both areas of the brain were lesioned, both the DMS and the DLS, mice became completely akinetic and just fell off the rotor rod at all phases of learning. So early and late and I guess naive. DMS lesions saw a significant performance decrease during early training that resolved in later trials, and the DLS saw lesions, DLS lesions saw performance decreases both during early and late trials. Conclusion they came up with was that the DMS is only required for early phases of training, while the DLS is required for late phases of training. Now, I don't really understand why they said this, considering the DLS lesion saw a decrease in performance during both early and late. They don't really go into this. I don't know why. I guess that's really kind of a flaw of the paper on their part, but that was a conclusion in which they stated in their paper. So we see that there is a difference in neural activity between the two regions, the DMS and the DLS, at the various phases of learning. To further support these findings, experimenters then took coronal slices and measured their invoked striatal field potentials. Results followed in the same path of electrode data, with the DMS being more active during naive and early phases, and the DLS being more active during or after significant rotor rod training. So for these coronal slices, they looked at spike amplitude, or basically how active these things were firing at given things. And again, it follows that paradigm of the DMS saw higher spike amplitudes, or a higher half maximum population spike during the early training and naive training, so basically early phases. and the DLS saw a higher amplitude or during the late phases of training. So we can see that there is a difference in neural activity during the various learning phases between the DMS and DLS. The authors of the paper then move on to trying to understand what exactly causes these dynamic shifts in both subregions. The DLS, you obviously see that nice increase in activity, while the DMS, you see that nice decrease. So you kind of got that like X thing going on. So they come up with two hypotheses. Either it could be a changes in synaptic strength or a changes in excitability. So they first try to figure out, is synaptic strength changing? And to do this, they do what they call a saturation test, where basically these two regions would be exposed to electrical stimulation in order to induce LTD. So basically the idea is that the stronger the synapse will have a more substantial synaptic potentiation. Upon stimulus to induce LTD, the stronger connections would have further to go in order to reach the floor. With this, we could expect the DMS to be stronger during early phases of learning and decrease with training, and the DLS to increase strength in training. And the results kind of follow this. So again, the basic idea is that a stronger synapse, you're going to have basically a lot farther to fall during application of LTD. And the results kind of found what they expected. It was found that more stimulus was required to reach LTD saturation in the DLS compared to the DMS after extended training. This is important as it suggests that rotor rod training results in synaptic. It is important as it suggests rotor rod training results in synaptic potentiation in the DLS. Now that's a pretty big statement because it really kind of sets the stage for a lot of the more later experiments. We shift focus away from the DMS later and focus more on this increase in synaptic potentiation of the DLS. This is the first mention of it in the paper. Next, they used a procedure called whole cell patch clamp to measure and compare the electric potential of DMS and DLS cells at various points of training. For this, they focused on the striatal medium spiny neurons. So whole cell patch clamp is a modified version of the patch clamp procedure where a very small micropipette is suctioned onto the surface of a cell. In whole cell, enough suction is applied to break the membrane of the cell and expose the cytoplasm. This allows for measurements of electrical potential and currents in the entire cell. So in this bottom right picture, I stole this picture, I snipped it from a PowerPoint. But basically, what you do is exactly what it sounds like. You just take a micropipette, you put it on the surface of a cell. In just normal patch clamp, they don't break the surface, and they usually just measure the electrical potential of like a certain area, usually around some of these membranes. But when you break this and expose the cytoplasm, you can then detect changes in electrical potential for the entire cell. Both the DMS, so what do we learn from this? So for both the DMS and the DLS saw an increase in EPSPs, or excitatory postsynaptic potentials, during early training when compared to naive training phase, which disappeared during late training. In addition, no changes were detected in paired pulse ratio, input resistance, or resting membrane potentials of cells of both the DMS and the DLS. Yin and the other authors go on to assert that these findings are proof that the DLS is undergoing some kind of long-lasting potentiation, and that there was no presynaptic changes. Now, if you're shaking your head wondering what a lot of these things are, especially paired pulse ratio, that was actually included on some slides that Dr. Farley never got to. When I went to speak with him, he kind of gave me a little bit of a crash course, and I'm going to attempt to explain it just as well as he did. So paired pulse ratio is basically pretty much what it sounds like, and it's basically a measurement to tell whether or not there is any presynaptic changes in terms of release of vesicles. So what happens is, and this is from the slides that he never got to, is you're exposed to some sort of stimulus that causes this first increase, this first reaction. And then if the second increase is larger, it indicates that there is some sort of change in the likelihood you're going to release them. So basically, you can either see an increase or a decrease. And basically, once this first stimulation comes in, you're having an influx of calcium, which allows the release of these vesicles. When you see a decrease, it usually means that the cell was primed enough so during that first burst, you released most of your vesicles and you haven't had enough time to reuptake them. An increase as seen in this one, this first action basically released vesicles but also primed the cell. So inside the cell there was an increase of calcium, and more increase of calcium results in an increased release of vesicles. In the case of this one, there was no change, which generally indicates that there is no presynaptic alteration of the cell, which would cause changes in synaptic activity. I hope I explained that well enough. It's not necessarily the easiest thing to go over, but I tried. The whole cell patch clamp procedure also indicated that the DLS has increased in NMDA to AMPA ratio after extended training. This was only observed here at the late stages of the DLS. Dr. Farley, again, gave me a little bit of a crash course on this and was actually very adamant about what this means. An increase in NMDA to AMP ratio does not necessarily mean that the number of NMDA increased over AMPA. Basically, there are three options of what that could mean, and he wanted to be very clear that it does not mean that NMDA has gone up. Basically, the first option is yes, it did go up. Another option could be that AMPA went down, or the third option could be AMD went up and AMP went down. AMPA went down. So, just to be clear on that, he was very, very adamant about that. This is kind of significant because generally the hippocampus, in terms of a lot of its LTD and LTD, use mainly AMPA for their baseline. NMDA receptors appear to make a contribution to the baseline in the striatum. So this is a nice differentiation between the hippocampus and the striatum. So we can see that the DLS undergoes some sort of preferential potentiation and has a marked difference in receptor ratios after extended training. This marks a shift in the paper from comparing the two subregions of the striatum to understanding how and why the DLS undergoes increased potentiation. So basically, the DLS has basically become a larger focus of this paper, and the DMS actually kind of gets left behind in the dust a little bit. These next few slides are actually, I'm going to go over pretty quickly because they don't really go into them very much. It's just basically this is what this is. So intrinsic excitability for the DMS and the DLS are basically the minimum amount of current needed to elicit a response. For the DMS saw a reduction in excitability after extended training. So basically it decreased, it was less easy to initiate a response with training, and this was the opposite for the DLS. So there's that. Threshold for psyching, which is exactly what it sounds like. It's basically the amount of current required to initiate a electrical response. The DMS had a slower stimulation requirement in the early training than the DLS, but increased dramatically after extended training. So basically, it needed more stimulation in order to elicit a response. Again, kind of going with the idea that DMS is more active during the early phases of learning. And the DLS had a high stimulation requirement in the early training, which decreased with training. And I think what is this called? I think Max thing cut that off. There was more to it. Look on the slides and the thing if it doesn't really matter, but if you're interested. And again, we kind of see this threshold for spiking. Kind of it goes over what I just said. The DLS required more stimulation in order to elicit a response in early phases, which decreased at late stages, and the opposite is true for the DMS. Yeah, it's pretty much exactly what everything we basically said. The DMS is mainly active during the early phases. The DLS is mainly active during the later phases. So we know there is a major difference between the DMS, which is mainly involved in early skill learning, and the DLS with late skill learning. The authors then wanted to examine how the direct and indirect pathways might be different in these subregions as well. Both direct and indirect pathways are known to be able to induce LTP in the striatum using different mechanisms. Specifically the direct pathway, which involves the substantia nigra, primarily utilizes D1 receptors, while the indirect pathway, which involves the external globules pallidus, mainly D2 receptors. Now, to be clear, both the DMS and the DLS both have these D1 and D2 receptors, and they are both able to use both the direct and indirect pathways. What these experiments are trying to figure out is do these subregions have a preference for the direct or indirect pathway? Just to be clear on that. Cool beans. Anyway, so and as indicated in the last section, there was found that DLS undergoes some kind of long-lasting potentiation after extended training. The idea here was that these two pathways may somehow play a role in this. To see if there was any significant preference in the DLS for one or the other, they used D2 green-enhanced protein mice to examine any potential differences. So basically, as D2 receptors showed activation, they would basically light up, which you can detect. There's also somewhat surprising in that D2 receptors showed a much greater potentiation in the DLS. This indicates that the indirect pathway becomes more engaged than the direct pathway as a skill is automated. Additionally, through the measure of receptor binding, the team discovered preferential expression of D2 receptors for the DSL. They have D1 receptors, but they were at similar levels to that of the DMS. So in short, the DSL has a higher ratio of D2 receptors, and they are more actively engaged during later phases of skill learning. So basically, what they're trying to say is that the DLS has more D2 receptors, and the D2 receptors are preferentially activated, so the indirect pathway is used more in the DLS during skill automation. So that being said, they were kind of curious that if they somehow blocked these receptors, how would performance on the rotarod change for these mice? And it was kind of interesting and not really understood. But when both the D1 and D2 receptors were blocked, animals completely became akinetic during the early naive trials and just fell right off the rod. But strange enough, and I didn't add it here into the thing, but I have a video to show this. During the late stages, they somehow were able to automate the skill and were able to stay on the rotorod for, I don't think it was a significant amount of time, but they were able to get on it. When just the D1 receptors were blocked, similar effects in the early trials of the both. However, after extensive training showed no skill handicap. So they were not able to at first get on the rod in early trials, but then they were able to walk on it, similar to the D1 D2 blocked. When just the D2 were blocked, performance was affected at all stages. And they seem to suggest, and they mainly focus on this D1 block thing, they kind of, unfortunately, ignored both the D1 D2 block and the D2 block. They say that this suggests that D1 receptors or the direct pathway activation is only necessary for early phase, and activation is no longer necessary for performance after extended training. So I have some videos showing mice with both the D1 and D2 block at the same time. And it's kind of interesting. It kind of raised some questions with me, and I'm kind of curious what you guys might think about it. But this mouse has been blocked by both D1 and D2 receptors, so basically he has not much activation of his direct and indirect pathways. He's placed on the rotor rod. It is spinning. He is a bump on the log. He's gliding. He does not care. And he falls. So there's that. And that is during early phases of learning, early trials. And again, I have to find this one. I'm terrible at this. So this is after late training, or during late training. Same mouse, both blocks, but he's all of a sudden able to automate this task and is able to stay up on the rod. They don't explain this. Again, another flaw of the paper. They kind of ignored this and preferentially looked at the D1 block and gave them kind of results that were kind of congruent with what they were expecting. They don't really address this. Interesting, but kind of sad too, because that was really interesting to me, how a mouse could not be able to practice and basically skip that early phase and still somehow go into the automation of it. Okay, no, come back. Okay, so there's the videos. So the conclusions, and I don't really have any notes for this. It's basically exactly what it says on the slide. Data supports the idea that with excessive training, the potentiation of excitatory transmission on medium spiny neurons of the DSL occurs predominantly in D2 or the indirect pathway receptors. As DSL experiences increased potentiation, D1 receptors become less dependent on activation. So, like I said earlier, we kind of shift our focus away from the DMS to the DLS. The DLS basically uses the indirect pathway more predominantly, and the D1 receptors lose or decrease in activity as as you train more. So we're on the home stretch. We're almost done, I promise. These slides are actually going to go pretty quickly because a lot of it's basically just overview of the entire presentation as a whole. So basically what we found is that we have found evidence for region-specific plasticity of the DMS versus the DLS. So that is your basic comparison of early phase of training being primarily involved with the DMS and late phase of training primarily being involved with the DLS. And we also have evidence for pathway-specific plasticity, D1 versus D2 receptors. We know that D1 receptors are engaged or D1 receptors, direct pathway receptors, are engaged in early training, and as training increases, as training is extended into the late phases, their activation decreases. And we know even more information about the D2 receptors, the indirect pathway receptors, and that the DLS preferentially has more of them and is utilizing them more. So we know those two things. This is basically exactly what I just said, but it also points to some problems that the researchers said with the paper. They don't know how long-lasting potentiation the DLS relates to automation. Basically, when we are automating a task, are we facilitating things? Are we facilitating the actual actions, or are we blocking those? Or blocking like competing actions? That's basically how they put it. In addition, it's unclear if DLS potentiation is accompanied by structural plasticity. So basically, what they're meaning by that, and I use that a lot, they aren't sure if there's an increase in number of spines in the synapse. They weren't really looking or examining that. They also said that's probably the next phase in which they want to go into. But that is that. And this is really, again, exactly what I've been saying earlier in terms of the direct pathway, mini-spine neurons, DSL, preferentially, blah, 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 blah. But there are some interesting implications that they add to this. D1 receptors are thought to be required, or thought to require increased levels of dopamine to activate compared to D2 receptors. When we see D1 receptors are just involved with the early phases of early training phases, this makes sense. Novel tasks are more likely to result in a release of dopamine, which slowly decreases as a task becomes less novel. And this has some implications that kind of jump a little bit, but it kind of makes sense too. This might result or have implications for something like Parkinson's disease. For Parkinson's disease, you lose a lot of the ability to have directed motion. So basically, with decreased levels of dopamine, levels D1 receptor activation will logically decrease as well. So basically, this might be the voluntary movements, but Parkinson's patients' automatic movements are the last to go. And since D2 receptors require smaller amounts of dopamine, this might kind of give a reason why Parkinson's kind of spares those more automized movements later. And that's really the end of it. This is where I was going to ask you questions. You're more than welcome to email me questions. My email is in the class roster. Feel free. I mean, I'm more than happy to answer what I can. Questions for you. Don't know how I'm ever going to get these. Basically, I've already mentioned these throughout the slideshow, but I was kind of curious what people thought about that. In the videos showing the D1 and T2 receptor blockages in mice, how is a mouse able to automize the run-around movements without being able to efficiently practice it? To me, that is bizarre. If we're defining the skill learning in terms of two phases, how are they able to automize a task or at least do late-stage training if they are unable to practice it? Is it some sort of delay? I don't know. I was curious. The experimenters in this paper didn't know. I was just kind of curious what you guys might think about that. And I think, again, I've also mentioned this, when we automize skills, are we suppressing unnecessary and competing motor programs or facilitating automatic motor programs? I don't know how you would do that, or how you would try and figure that out. My question to you is going to be, how would you test for this? Or how would you try and figure this out? I mean, I'm not very sure. But we're at the end. Thank you all for watching this. I apologize for me having to look down a lot. OBS tends to not like Macs and uses a lot of CPU, so I keep getting error messages in the bottom saying, hit OK or we will shut it down. But we're done, and I hope everyone's presentation on Thursday go well, and it's been a pleasure to have you all in class. So thank you very much.